this is a group of people who know each other, I guess, primarily. So so there's not a lot of need of our introduction. So I think we should go straight into it and give you a seminar. And it's 45 minutes, isn't it? And 15 for questions at the end if we got a chance. How about that?
Sounds good. Yeah.
Great. Thanks, Aiden. Take it away.
Okay. Thanks, David, and thank you to everyone else for joining in this morning. So, and I guess as part of the analytics scene, and we're trying to have some seminars like this and to share various ideas and thoughts. And my own today, there are around wearable sensors. So I guess I unashamedly am picking the area that I invest most of my time in, and then also discussing very much around reproducible machine learning and of movement behaviours and wearable sensors. And I guess why wearables? First of all, well, I guess right now, today in the UK, there are 8 million people walking around or sitting around who have a commercial wearable such as an Apple Watch or a Fitbit. And these type of devices collect a wide range of information and physiological traits, from simple measurements of physical activity or sleep to also to physiological more silent measurements such as heart rates, heart rhythms, and blood pressure monitors, etc. So there's a lot of people walking around with this huge wealth of essentially what's largely unexploited information. And the big appeal of using these or of trying to exploit information from these devices is that it really does offer the potential to transform our understanding of the causes of, consequences of, and assess treatments for a wide range of disease outcomes. And to give one example of that, if we consider the association between physical activity and heart disease, we might just naively think that, well, surely more activity is better, but there's been a lot of essentially dispute in the literature over what we call what is the dose response association between physical activity, shown here on the x-axis, and coronary heart disease, sort of the event rate shown on the y-axis. And we can see this effect in the Million Women study, so a large self-reported study, a longitudinal follow-up, that appears to indicate that more frequently engaging in strenuous physical activity or any type of physical activity might actually be adversely associated with coronary heart disease. So but other studies have indicated that higher levels of activity are beneficial, so there's genuine uncertainty over the understanding on a fairly basic relationship, such as between activity and cardiovascular disease. And that's because in the past, epidemiologically, we've relied on self-reported measurements of activity, which are crude and prone to measurement error. And just to highlight that, the national population representative surveys took place in the US and in England about 12 years ago, tried to assess the amount of adults who met physical activity recommendations. And you see, when you ask people via a self-reported instrument or try and directly measure it via an accelerometer, we get very different estimates on the amount of adults who meet these guidelines. Which measurement is correct? Well, there's never a perfect measurement instrument, but certainly the accelerometers are much more strongly associated with gold standard measurements, physical activity, energy expenditure, and also there's some emerging evidence from smaller cohorts showing that objective measures of activity and fitness are more strongly associated with mortality than a self-report. So in that regard, there are still a number of unanswered questions around physical activity, and I could easily apply these to sleep or to circadian rhythms or to other behavioural attributes that have been traditionally measured via self-report. And some key questions that I've highlighted is, well, how strongly is wearable measured physical activity associated with incident cardiovascular disease? This could also be neurodegenerative diseases or cancers. What types of activity are associated with lower CVD incidence? And then does activity causally lower cardiovascular disease, i.e. is it more important to be active or to have the capability to be active? And also, are these measurements trustworthy? So be talking about around this aspect of reproducibility and trustworthiness that I know is important to a lot of us in the applied analytics theme. And then finally, how can we embed sort of these new measurement instruments into clinical trials? So I'll discuss some of these aspects over the next 35 minutes or so. And so what I should have said at the start is if anyone's got a burning question, do feel free to interrupt me as well. So what we would really like then to answer many of these questions is a longitudinal study with a wearable measurement on individuals, and we follow them up for decades for clinical outcomes. And in this regard, we're obviously very much blessed here in the UK then to have the UK Biobank study. Many of us will be familiar with it, and the most interesting aspect for today's talk is the physical activity monitoring component. In the graphic of the sort of cartoon person, that's the blue device placed on their right wrist. Because in the UK Biobank we have distributed wrist-worn activity monitors, such as the one shown in the top left, to 100,000 participants. And they were distributed about 2013 to between 2013 and 2015. So 103,000 people agreed to wear a device and returned back data as well. And one of the first challenges we have is kind of a very applied analytics type challenge is, well, it measures a person's movement as they go forwards and backwards, side to side, up and down 100 times a second. So for each individual in the UK Biobank, we have around 180 million data readings on their movement behaviours over a 7-day period times 100,000 people. So there's a computational challenge in dealing with the volume of the data, and an even greater challenge in trying to extract clinically meaningful measurements. As one imagines, you've got tens of millions of data readings. There's almost an infinite amount of phenotypes that one could extract from this, but we want to extract the best and the most valid phenotypes that we can. So as part of the Biobank exercise, then we convened an expert working group of researchers in this space across the UK, and through a process of expert consensus then came up with this particular processing pipeline of ensuring devices are calibrated. So essentially, if I had two devices and put them on the table beside each other, they should give the same output, but often they don't, so we need to calibrate them. And this is very similar to the imaging domain, then, where you need to try and calibrate across scanners. As well, we need to, a component then unique to activity monitors, we need to remove noise and gravity. And additionally, then, when you ask a person to wear a device, not everyone will wear the device all the time. So we need to detect when the device is being worn and not worn. And finally, then, adjust our metrics based on the times of day that people wear the device. Also, it's important to try and determine what's the minimum amount of time that a person needs to wear a device for us to be somewhat confident that we've got a stable measurement of their physical activity status. So we worked closely then through all these issues over a period of a couple of years. and came up with this particular method then to infer essentially the overall amount of movement a person was doing, and this has been validated then against doubly labeled water, which is seen as the gold standard measurement of physical activity energy expenditure. And the neat thing about this in a time series is we can then construct these 24-hour models of physical activity and movement behaviours in 100,000 UK Biobank participants, whereby x-axis is the hour of day and y-axis is mean acceleration, so essentially a proxy for overall activity status, whereby higher values indicate a person is more active. And we can see, for example, just descriptively that older participants, and both older men and older women, are much less active than their younger counterparts in the afternoons and early evening time. But as I said, this is sort of purely descriptive work, and I'll be showing longitudinal associations soon. And furthermore, with the UK Biobank, there's a question about, well, how, if I ask a person to wear a device for 7 days, how good a measurement am I getting of how active they are in general? So how stable is a 7-day measurement? So we haven't quite published this yet, but we've asked around 2,500 Biobank participants to wear a device for 7 days, but at 4 time points through a year, to try and see then how stable is any one 7-day measurement versus the average of the other 3 7-day measurements. And we can see that there's for the overall activity attribute on the left-hand side, I'll describe the others as we go through the talk a bit later. We can see that the within-person correlation is about 0.75 to 0.8. So essentially the take-home message in this is that a single 7-day measurement gives you a lot of, or can represent a lot of the variability of a person's physical activity status at least. So that makes us feel quite confident using a 7-day snapshot for associations and with health outcomes. And because it's now over 5 years since individuals first wore these devices, we can begin to do longitudinal follow-up. So this is a really nice paper then by Tessa Strain at Cambridge that was published back in September, that after 3 years of follow-up, associating the physical activity variable, which is on the x-axis here, with all-cause mortality, where the hazard ratio is on the y-axis, we can see there's a very steep decline in incidence of all-cause mortality as one is more active, as measured by the accelerometer. And there's no hint of kind of a dip up on the right-hand side for adverse effects for people who do more exercise. So this is providing some really neat insights then into the dose-response association. And at Oxford, then we published a complementary paper then just back in September looking at incident cardiovascular disease. So this was with 5 years of follow-up and around 3,500 CVD events. And again, very similar to the association of all-cause mortality, we're seeing essentially what looks like a linear, or at least curvy linear association between accelerometer measured physical activity and the incidence of CVD. And the other noteworthy thing here is that in recent, for example, chief medical officer guidelines on how much physical activity people in the UK should be doing, it's the same for the US Surgeon General guidelines in the US, it's been assumed that those who are most active versus those who are least active have around a 20% reduction in their CVD incidence. But what we're finding here with the device-based studies is that this, I guess the hazard ratio is probably more so indicating around a 40% possible reduction in CVD incidence. Now, of course, we've just got 5 years follow-up here, so we'll have to keep a careful eye on this, but it does appear that the association between physical activity and cardiovascular disease is much more strong than we previously thought using the self-reported measurements of physical activity. And of course, what I've shown so far is just broadly overall levels of activity as measured by a wearable. But as mentioned, then these devices capture really detailed, kind of exquisite measurements of people's physical activity and sleep status. So what types of activity then? So can we get at some behavioural attributes? And the intuition here is that we've got these measurements from a wearable on the left-hand side in terms of the accelerometry, but can we identify patterns or signatures within this that indicate if a person is sitting, or if they're walking, or if they're driving, or doing some other activity of daily living that we believe is important? And people have tried this in the past, but there have been two key limitations to this. The first being that most of the computational models being developed were perhaps being developed by people from non-computation backgrounds. And secondly, more fundamentally, the models have been developed and trained in laboratory settings, whereby I might follow someone around for an hour with a clipboard telling them to do a series of activities for an unrealistic amount of time and an unrealistic sequence, and therefore I develop an unrealistic model. What we really need is free living data sets, whereby models can be trained in the environments in which we intend to deploy them. So what our own group then at Oxford have used is a wearable camera type device that looks something like this here, and it takes first person point of view images. And so essentially what we've collected is a training data set of around 150 individuals or thereabouts, who are wearing a camera so we can capture exactly the behaviours they're doing over a full 24-hour period. And also we've asked them to concurrently wear a wrist-worn accelerometer, the exact same as used in the UK Biobank study, worn on the same wrist, so following the exact same protocol. And this enables us then to develop essentially a really robust way to validate how well methods work in free living environments. So from the accelerometry data, then we've developed a two-stage classifier, then a random forest and hidden Markov model. So I'm going to work from the bottom up to the top in this slide. So we first of all take a 30-second window of raw accelerometry data. We then extract a range of time and frequency domain features from this to make a preliminary and plug those then into a random forest classifier to make a preliminary prediction on what behavior an individual is engaged in for that 30 seconds. And of course, we're dealing with time series data, so it's really helpful then to apply a hidden Markov model over that. So the intuition being, if a person is, I don't know, an extreme example would be sleeping, bike riding, sleeping. It's unlikely a person would offer a 30-second bike ride in the middle of the night. So instead, the hidden Markov model smooths over the erroneous classification. How well does this work? Well, as mentioned then, so we had around 2,500 hours of free living behaviours in around 150 individuals. From the camera, we identified 230 different types of behaviours, which is perhaps not all that helpful. So instead, we collapse it then into 4 major categories as shown on the confusion matrix just below, namely inferred time spent asleep, time spent in sedentary behaviour, time spent in light activities, and also time spent in moderate to vigorous activity. And we're very pleasantly surprised on how well this works. So with an accuracy of 88%, or my own preferred metric then for this is the kappa score, which takes into account the distribution of the classes. So we have a kappa score of 0.8. So not perfect, but a lot closer to perfect than random. So this does work, I think, really quite well. And we've tried many different types of models over the last few years, and it is frustratingly hard to try and beat the performance of a random forest classifier that's smoothed over by a hidden Markov model. And the key thing for us then is that the tools and methods that we develop are made available then to the wider research community. So on the left-hand side, I've just taken a screenshot from all our accelerometer processing tools are made available on GitHub for the wider research community to use as they actively do. And also any derived variables that we extract from the UK Biobank, and any new phenotypes we developed are put back into the UK Biobank showcase, again, so researchers both nationally and internationally can use them. And for people, probably particularly in the applied analytics community, who are interested in machine learning, up to now, for people who work in human activity recognition, just the data sets have been pathetically small. So we are imminently about to release this 150-person data set. It's we've got all permissions in place, and it's just now a matter of getting it hosted up on the relevant gateways. So it should be available at some point over the next 2 weeks for people to use in the community. And we're not just going to have a dump of files out there. We've also prepared some code to go along with this, essentially a set of tutorials to go along with the data through, which we believe will be really important for training of researchers across the applied analytics team and beyond as well. So then, having deployed this measurement of essentially behavioural measurement to the UK Biobank data set to try and infer time spent sleep, sedentary, light activity, moderate vigorous activity, it would be tempting to run off and do health association analysis using traditional models. If, for example, I want to interrogate whether an hour extra of moderate to vigorous activity, how that might be associated with, let's say, cancer. But I can't really use a logistic regression model because it assumes a unit increase in moderate to vigorous activity, okay, but then also assumes that everything else is held constant. But I can't add an extra hour to time spent in vigorous activity, because then I violate the very hard constraint that is a 24-hour day. So instead, I need to take that time out of the other behaviours, and it's usually not a straightforward kind of substitution to, oh, this sleep an hour less, and therefore that will magically create the time and to do all moderate to vigorous activity only. So instead, it's really important to use methods such as compositional data analysis that essentially take these considerations into account. And Rosemary Walmsley at Oxford has been doing some really brilliant work in this regard. So using the machine learned behavioural phenotypes and compositional data analysis, Rosemary then has shown the association and of substituting time between behaviours and how that's associated with incident cardiovascular disease. So again, this is around 5 years follow-up, around 3,500 incident events cardiovascular disease. And I'll talk you through the graph on the top left-hand side. So the x-axis then is kind of substituting more time spent to sleep versus more time spent in sedentary behaviour. And then the y-axis is the, so that's while holding some of the other behaviours a constant. And then on the y-axis, we've got an estimated hazard ratio. So the intuition of this graph to the left is that as one replaces sleep with sedentary behaviour, there's an apparent detrimental association with respect to incident cardiovascular disease. And then looking through the other graphs, we can see the trade-offs between other behaviours. And what's striking then is that moving from any behaviour type to doing more moderate to vigorous physical activity is associated quite strongly with lower incidence of cardiovascular disease. And of course, such associations, as I highlighted a few minutes ago, can still be prone to residual confounding and reverse causation bias. So to then get around that for causal inference, what we would really like to do is combine, for example, the activity measurements and phenotypes with genetic approaches to aid and enhance causal inference. So using the data in the UK Biobank, this paper here then shows the genome-wide association study with various machine-learned behavioural phenotypes that were extracted in the UK Biobank. So we found 14 SNPs, and for example, the overall activity variable was around a heritability of around 20%. As if one combines all the genetic variants together, can explain around 20% of a participant's physical activity behaviours as measured by an accelerometer. And as with other work done in our group, then the GWAS summary statistics are publicly available on the GWAS catalogue, so that others then can build on top of and use the summary statistics from this work. And this, first of all, gives us some insight into the biological relevance of accelerometer measurements, showing probably unsurprisingly for a behavioural trait, there's a very strong role to play for the central nervous system, and lots of brain tissues are implicated in various behavioural measurements from accelerometry. And as well, we can use these methods to, or these genetic variables as instrument variables in Mendelian randomization analysis. And just very briefly, for those who are unfamiliar, then Mendelian randomization is somewhat analogous to a randomized control trial, whereby the alleles are randomly assigned them during the process of meiosis. So instead of being randomly assigned to a physical activity intervention, you're randomly assigned to genetic variants that infer or that tend to lead to people being a little bit more active. And some emerging MR, Mendelian randomization, then using the phenotypes we've developed in UK Biobank and the genetic variants associated with those. So using that human genetic information, there is evidence suggesting that increased overall activity is causally associated with reductions in diastolic blood pressure, odds of being diagnosed by hypertension, and also odds of being diagnosed with depression. Currently, there's no evidence for, or perhaps more accurately to say, we're currently underpowered, I think, to detect associations with, for example, coronary heart disease, MIs, ischemic stroke. Although the p-values are very far away from suggesting that there's even hope of evidence for an association, I think that's something we really need to unpick much more carefully over the coming years. Although I would like to highlight, for overall activity, we have got just 5 genetic variants associated with that. So we're somewhat operating with our hands tied behind our back. This was a paper published then last year looking at Mendelian randomization of various traits with, I believe, sorry, I should have highlighted this with cardiovascular disease. And as you can see, when comparing the physical activity instruments compared to all the other behavioral types, we're just dealing with a smaller subset of genetic instrument variables. So as big and as wonderful as the UK Biobank is, we really need more and larger studies with wearable measurements and genetics as well. So hopefully in the future we can enhance the size of the UK Biobank accelerometry data collection and also bring in other cohorts as well. And I'll discuss that just a little bit later. And I've talked a lot about these behavioural measurements of physical activity that are inferred using machine learning. Many of us across the applied analytics team use machine learning in one flavour or another. And there's, I guess, then a question around how trustworthy are these methods? Because the background to this is that there certainly are question marks over the robustness of machine learning methods. In the field of imaging, for example, is a really nice exemplar then published in Science a couple of years ago in the field of dermatology, then trying to identify benign versus malignant skin lesions. And on the left-hand side, we've got an original input image. Then we can see the machine learning model is very confident that this is a benign growth on the skin. But if one perturbates that with some just adversarial noise, and as you and I look at the image and compare the original image on the left and the adversarial example on the right, there's no apparent differences to the human eye, but it really does upset the machine learning method then, which suddenly becomes highly confident that this is a malignant skin growth. So we need to be careful that our models are very robust. And as well, there's very large concerns around reproducibility of machine learning models in healthcare. And the Turing Institute have created this nice 2x2 table up on the top right-hand side, talking about what is reproducible versus replicable versus robust versus generalizable. And really our lowest bar to entry is reproducibility, but we're very far from achieving that as a field overall. Whereby our reporting standards are they're not standardized essentially. Sharing of data is difficult, and I think for some very unique reasons to the health domain versus others. But we don't really have much excuse then for not sharing code, which is becoming more widespread now, which is really encouraging, but still very a wide range of concerns around the challenges to reproducibility of machine learning models in healthcare, as indicated in the JAMA viewpoint on the bottom right. And to one last slide then to beat that drum with is a paper published just 2 months ago in Science Translational Medicine then that evaluated 511 scientific papers across several machine learning subfields. And the take-home point then from this is that, well, first of all, the authors here then looked at 4 different attributes, where A1 in these graphs refers to whether the code is made available, A2 is whether they use a publicly available data set, the B1 talking about just whether basic standard statistics are reported then on models, and then finally seeing whether models are compared across multiple data sets. And 3 fields were compared then, so machine learning for health, which is on the x-axis along all of those, and it was compared then against natural language processing, computer vision, and sort of general machine learning. And essentially the machine learning field is underperforming the other fields by quite a substantial margin and quite noticeably as well. So as a field overall, we do need to improve our reproducible practices. And in this particular paper, they had suggested some ways forward. And I have shamelessly then picked 4 of the areas in particular, because that's what we're covering for our implementation project on reproducible machine learning, namely a need to improve reporting guidelines, a need to improve sharing of data, a need to improve sharing of code, and I think particularly for us in safe haven environments, well, that can be quite difficult at times. and also a need to improve training and practices to improve the culture or that machine learning, but reproducibility is just something you do as standard when developing such models in health data science. And to give a little update on where we're at with the reproducible machine learning project, a really key part of this is that we work together then with patients and members of the public, and very surprisingly then to us, we advertised a flyer on the right trying to recruit a PPIE group, and we received 80 expressions of interest, and we were only hoping to recruit a panel of 10 to 12 people. So we had to spend an extra couple of months then trying to figure out how to appropriately then filter out this to appoint a group of 10 to 12 individuals. And we'll have our first meeting with this group to really get into the nuts and bolts of the project and get people's input. We'll have that first meeting next month. Around reporting then, this is led by Gary Collins at Oxford, and again, just highlighting that reporting is very substandard at the minute. This is a systematic review of machine learning models used for risk prediction, and even simple things such as study dates, sample sizes are not being correctly reported. So Gary and his team then are leading the new tripod AI reporting guidelines. The first round of the Delphi survey has been launched, and Gary and his team are currently going through this information, and there'll be a second round of a Delphi process. So hopefully at some point over the next year then we'll be in a position then to publish these tripod AI guidelines, and there'll be a lot of interest in the major journals in this, where, for example, the Lancet then invited Gary to comment on the reporting of AI prediction models, and all the major journals are keen to adopt the tripod AI guidelines when they are created. Around sharing of data, we are working closely then with others in HDR UK around synthetic data sets, which I believe will be very much of interest to the wider HDR UK community. I often think of my wife and this, who does some epidemiological research with the NHS Scotland data, whereby she had to wait probably, I know, around a year to get access to the data, and then took her another 8 months to figure out how to appropriately analyze that data. And it would have been ever so helpful to be able to have access to it while waiting for all the approvals, to have access to a synthetic version of the data set. So one can then get a feeling for what the data looks like, how messy and noisy it is, and begin to prepare analysis pipelines around that, so that then when approved for access to a data set, a researcher is ready to run on this. So we convened a workshop around synthetic data, only expecting a handful of people to come along, but we had 160 attendees. So I think it's a big appetite across the health data science community for, I think, more for a greater variety of synthetic data sets to be made available. And of course, they have to consider some of these key attributes that I have listed on the left-hand side. With safe havens as well, we're working closely with Catalina and Vallejos, who is also based at the Alan Turing Institute, and our exemplar here then is working with the SPARA exercise. So I never remember the full acronym of it, but essentially it's a risk prediction for readmission to hospital, and working closely with NHS Scotland data and developing machine learning models in these restricted data safe haven environments. And a paper is currently under review on this. And essentially there are some hard and soft obstacles to operating in a safe haven environment, but we have some potential solutions to those, and as well the wider move towards trusted research environments all have computing more integrated into that will be very helpful. I'm going to take a little drink of water. So I'll soon wrap up because I just noticed the time. Training is very important. So at this year's summer school, we will have hands-on training on reproducible machine learning for the students using the human activity recognition dataset I talked about a little bit earlier in the talk. So the people working with open access data will give them code to help train and also be bringing in some of the aspects of the reproducible machine learning guides. And then additionally, we're working with the UK Biobank, which is moving towards the Amazon Web Services platform. We'll be creating training material in that, again, to around reproducible machine learning of time series sensor data to help the wider research community. And I will I'll briefly just talk about clinical trials and then wrap up. So I said at the start, there's a huge potential to assess which treatments might work for various outcomes. And accelerometers have been used as the primary endpoint in trials. Here's an example of a heart failure trial that was looking at the oral nitrates, and the primary outcome measure was accelerometer measured physical activity. And building on this, the FDA have created a set of guidelines around guidelines and recommendations around use of mobile devices in clinical trials. And there's a huge, I think, opportunity for us to interact then with clinical trialists and to embed these ultimately more accurate measurements of patient relevant endpoints, such as particularly around wellness. And there are large EU projects. Actually, both of these are led out in Newcastle, both are around £40 million or euro projects that have multiple partners across industry, academia, and the EU as well. So I think it's an opportunity for us in the applied analytics team and to work proactively then with such groups who are trying to create clinically validated measurements from wearable sensors that can be used in regulatory trials. So this is my second last slide. From all of what I've discussed, where are we going next? I just wanted to highlight some areas that undoubtedly I'll be featuring more in talks over the coming months, but the work just isn't quite ready to share yet. But our own group in Oxford are working on deep learning methods for CVD risk prediction, around self-supervised learning to identify sleep stages as validated against polysomnography and the gold standard measurement of sleep, multi-stage machine learning methods to identify even a simple, or what sounds like a simple, but it's actually quite hard to measure attribute of steps, particularly in recovery after surgery. Then we're also working on the interpretability of deep learning methods with respect to rheumatoid arthritis, and developing recurrent neural networks with a target area of Parkinson's disease. Many of these are aiming to enhance the UK Biobank resource for the wider health data science community. And this is my last slide. In terms of where we need to go next, I think we need larger and more diverse wearable data sets. For example, this year then we're the ELSA study are collecting accelerometer information on a population representative sample of older adults, around 4,000 of them. And we've got sister studies in China on up to another 40,000 individuals that we're collecting wearable data on. There'll be certainly a lot more emphasis in future then on new phenotypes, such as the sleep stages, new analytical methods, hopefully working together with you and others across the applied analytics community in HDR UK. And of course, we need new collaborations and partnerships, not just across academic disciplines, but also working together with industry, whereas at the start I said there's 8 million people walking around with these devices, and largely this information has not been looked at all with respect to helping improve our understanding of health outcomes. And then finally, if you do know of anyone, we have a couple of positions at Oxford that will be very soon advertised around machine learning and genomic discovery. So please do make others aware of this then too. And with that, I'll stop and thank you very much for your attention.
That's great. Thanks, Aiden. Would you like me to handle the questioning thing for you, so you can relax?
That'd be great, Dave.
You can relax and answer the questions rather than worry about handling. So we've got there was some questions that came up in the chat as you were going through. I think people were probably too polite to shout out in the talk itself. I don't know how many of them actually subsequently got covered. So when you were talking, so I'll just go through quickly. So Albert was asking about the recommendations for physical activity was self-reported or measured, but I think the answer was both in different ways. I don't know if you want to say anything about that, or this is kind of a raw data question. There's another one related from Anna Barnes, asking how the classification was done to running, cycling, walking, that sort of thing. What's that then? So I think this is a kind of generic measurement bunch of questions. I don't see anything you want to say about that.
So first of all, yeah, to Albert's question, then, so are the recommendations for physical activity based on self-reported or kind of more directly measured data? It's currently all based on the self-reported guidelines. It's a little bit of a tricky one that, so I think the wearables are really good to generate the evidence base for understanding how important is, activity for cardiovascular disease or cancer or mortality. Although people, as we've shown, that are quite bad at estimating to the device, say you do X amount of activity, people are very bad at estimating what X should be. So probably our guidelines might still continue to be based on the self-reported data, because if people then report they do, Y amount of activity, and that's associated with favorable outcomes. It might be easier to tell people to aim for Y rather than X, because Y is what they think they'll do anyways. But I think the rationale then for emphasizing activity with respect to various outcomes, we all really ought to be using the measured outcomes for that, or the wearable measured. And I think I noticed Anna's question then is, does my Fitbit know if I'm doing running or cycling or walking? It tries its best to estimate that. It's certainly not going to be perfect all the time, and I think what we really need is some independent validation studies in free living environments for those. So possibly good, but we don't exactly know, and I think we need better validation studies on that.
Okay, just going down through the list. So you can see it as well as me, Aiden, actually. The Omar Khan's one about whether you're factoring in things like BMI and so on. The issue being how do you disentangle the activity versus just general health risk?
Well, thank you, Umar, for the question. So when looking at the association between physical activity and, for example, cardiovascular disease outcomes, we do take a range of kind of your standard confounders into account, such as sex, ethnicity, area deprivation, diet, but we don't take BMI into account because it's what we call is on the causal pathway. So essentially, when one changes the activity, that changes a person's BMI, which in turn is also associated with activity, because it's really hard to disentangle that. So often we don't include what we call these variables that are on the causal pathway.
Good. And then there's a bit of dialogue. Actually, it was Manuel Trucco was initiated it about. So you're in love with random forests, but how does it compare to things like convolutional neural networks? That kind of all of that deep learning jazz.
And I see that my brilliant research colleague at Oxford, Xing Chan, and has answered some of Manuel's questions much more ably than I would. So Xing has led the interrogation of a lot of other methods, and such as CNNs and LSTMs. I noticed Xing's mentioned there too. And they do struggle to beat the random forest. I think they might be slightly better, but not noticeably so. But yes, at Xing, if anyone's particularly interested in deep learning of time series data, Xing is definitely the person to ask on that.
All right, there you go, a plug for Xing. And a comment from Will Whiteley about you're talking about NLP versus learning in broader in medicine. So I think that's just a comment, Will, unless there's anything you want to say. Don't know.
It was just to say, Aiden, that we've just done a similar exercise in NLP, and really we haven't found it much better than in machine learning.
Dun, dun, dun.
Unsurprisingly, perhaps.
Yeah.
Thank you, Will. You've given me another paper to read there too, and that's very helpful. Thank you, Will.
Great. And then another one from Anna about building in quality management and patient safety into the design of these things. How do we do that? You'll tell us. There's an easy answer to that.
Great question, Anna. How do we build quality management and patient safety in the design of these tools? Oh, well, I guess we're hoping to learn some of that in this project. This is a pretty big nut to crack. And I suspect, even within the remits of just a single project in this space, that we will struggle to do so. There's certainly incredibly important attributes. With the reproducible machine learning project, the minute we're just trying to make sure that a method someone proposes can actually be reproduced by someone else. So that's kind of our lowest priority entry. And as within the project, we will look at whether it's robust to some noise or perturbations, these methods. But ultimately, I think before probably any of these methods be brought into sort of day-to-day patient care, they really ought to undergo, I believe, assessment and evaluation by randomized controlled trials. I think cluster randomized control trials could be a nice way of evaluating these methods. But before going to a randomized control trial, we would like to have a method that we believe gives us the best chance possible of having a positive outcome. And I think we need to get the robustness right before we get there. And I know I've given a bit of an invasive answer, but it's a pretty hard nut to crack then about ensuring patient safety in the design of these tools.
And then Omar Khan is asking about data standardization for robustness. So would that help? I suppose the general answer is yes, it would. So I think it's a practical question. I mean, in practice, is that something that would be a good thing for the community to do?
I'm going to give an unhelpful answer to this. I'm going to say yes. And probably, Dave, you'd be the best person to comment on.
Me? Oh, I do. So the question is really would it help if we standardized our data more so that we could then do the validation robustness testing because it would give accessibility to more methods more easily. I think it's a practical engineering question actually, unless I misunderstand you. In which case, the answer has to be yes, I think. But then the question is, in practice, is this going to work? Because we need to standardize. Standardizing is a good idea, always good idea. But then you also need to be standardizing the computation so that computation will then fit to the standardized data in some predictable way, which is not a trivial thing. I think there is an issue for TREs. Actually, you touched on it, Aiden, when you were talking about being able to do that sort of thing. And as far as I know, there isn't. I mean, there are generic solutions to this, but you don't see them an awful lot in practice. I think possibly because it's you're into the specification world doing that. and you have to specify the algorithms and then turn them into a computation somehow. So sorry, I'm warbling away, and I don't want to dwell on that one.
Just very briefly, I did sort of just add one little extra thing then. So yes, it's something we certainly like to aim for. It is very challenging because even 2 research grade accelerometers that are commonly used, if then developing machine learning methods for those, the models don't nicely transfer from one accelerometer that measures at 100 hertz versus another measures at around 85 hertz. Since some of the input features of a machine learning model can change some of the predictions one gets from that as well, yet alone trying to think about a research grade device versus an Apple device versus a Fitbit device in the future. So standardization certainly is a goal for us to aim towards, but it's, as you said, Dave, certainly a non-trivial one to work towards as well.
Mm-hmm. Okay, then Gideon Giacomelli, ethics in machine learning AI. If you get an incidental finding, what do you do?
Great question, and thank you, Gideon. So I have to admit that I guess my interest, probably quite unashamedly, is how can we develop the best tools possible, and then I would like to work with people who are much more clever than me then to think about, well, what are the ethical implications then of the use of that tool. There is certainly a lot of interest in this space, and for example, around incidental findings, I guess, when in cohorts such as UK Biobank, there's the decision that generally incidental findings are not reported back to participants, but in other newer cohorts do take that into account. Of course, and integrating into clinical care, that's important, but I have to admit that I'm not the probably expert task on ethics and ML and AI, but there certainly are others, and I know I'm going to put a little plug for Oxford here, and so we've got a whole new AI and Ethics Institute that's been created in Oxford that had a pretty substantial donation of over £100 million. So it will be, I think, becoming more of a kind of a centre for action, and some of my colleagues there will be much better placed to answer such questions than me, but it's really important to raise these questions as well.
One from Pippa Thompson. Sorry, we're overrunning a little bit, so but is that okay, Aiden?
It's fine with me.
I'll try and rattle
okay.
reasonably quickly so people can get away, but it's interesting. So Pippa, how much do accelerometer measures improve prediction of CVD over heart rate monitoring? So you just did heart rate monitoring as opposed to?
Great question, Pip, and I'm not sure anyone's actually really looked at this in anger. So Xin Chan, who I mentioned, and also Ben Feekins and our team are looking at currently the question of how much do accelerometers improve the prediction over your standard Q-risk score that's used in day-to-day clinical practice. There's probably wearable heart rate monitoring. There's no real large studies of wearables out there currently with heart rate monitors that are longitudinally linked to clinical outcomes to properly answer that question. In the UK Biobank, we are in the process of collecting data from around 30,000 individuals with heart rate monitoring. But then to answer your question on CVD risk prediction, I imagine might be another 8 to 10 years before we're in a position to answer that. And how much do accelerometers improve prediction over traditional clinical variables? We've got a lot of hope, and we're just currently undergoing an external validation of a model that we have on that. So watch this space.
And Jackie MacArthur's pitched in with after Omar's question on the BMI issue. I don't know, Jackie, if you want to say anything about that. I think the intuition is, gosh, that might be really quite an important but you're welcome to pitch in.
Jackie may have stepped out there.
Maybe she's gone. Oh well, I think that's really a comment. And then Will.
So Will's question.
You can read the question.
That's okay, Dave. So Will's question, so looking to see how well accelerometry tracks FDA community outcomes as a work in this. So Will, it's on my eternal to-do list actually to reach out to you on this, because well, you do know my bias of using these devices, and I think they can be really good outcome measurements. So in hemodialysis participants, we have validated or correlated this with a heart failure questionnaire. But I think what I'd really love to do is integrate these devices as an endpoint measurement, probably in heart failure trials. It's the obvious area, and to then validate against standard instruments in that. And as well, we wouldn't, or I'd be very disappointed if an accelerometer or wearable correlated perfectly with one of the standard questionnaire outcomes, because even though they're the standard, they are very far, I think, from measuring what we really want to measure. So you'd like to see some concordance between the measures, but not too much as well, because when you believe the underlying reference standard isn't very good in the first place, you don't want to exactly match it as well.
Great, and I think this is the last question actually. It's all of the things below that in the chat were just thanking you actually. So but Albert's asking about, do you happen to know if there is believed to be any genetic correlation between activity and BMI, that some sort of causal pathway? Is there a genetic overlap, that kind of thing?
So briefly, yes, and a pretty strong genetic correlation between them. So it's, I can't remember the exact correlation coefficient off the top of my head. There is a strong correlation between both traits. And then to make it even more complicated, there is using the Mendelian randomization, activity lowers BMI, but then also BMI lowers activity. So this kind of really complex and bi-directional association between them that makes it very difficult to unpack. And that's why I love larger genetic studies of wearable measurements of activity to help really unpick that a bit more.
Good. So that's all the questions in the chat, and I see Rebecca put in a note to say we'll circulate the recording and all this sort of thing, so people can pick it up again if they want to. Is there any any final last question anybody wants to ask? You have to shout if you want to. You just shout it. I don't hear any shouts. So thanks, Aiden. That was a great talk, as usual. And thanks everybody else for attending and sticking with it, and enjoy the rest of your day.